So please, let's begin this session and try to be on time. Well, the first speaker for today is Professor Antonio Moretti. He is a research leader of the Institute of Science of Food Production of the Research National Council of Italy. Please.
Good morning, everybody. Thank you very much to organizer for the nice invitation. I will try to speak about some work we have done on the diagnostic tools for identify toxigenic Aspergillus and Fusarium species of maize by using different approaches from our institute or in cooperation with other colleagues from other groups. And I will introduce a bit, because I know that here not all are plant pathologists and or mycotoxicologists, so I will try to introduce a bit the matter of mycotoxins for those we are not in the field. They are products of the secondary metabolism of microscopic fungi. There is 25% of food, according with some data of FAO that were then recently confirmed by a nice review by Yestoy and co-workers that at global level is significantly contaminated by mycotoxin. They are still largely global health issue, ignored in some area. Mostly there is no the good efforts yet for the solving or mitigating the problem in the incoming countries, mostly in Africa. And the losses of products are enormous. Just only in US for the barley, the wheat and barley, the Fusarium mycotoxin cause a loss of around 3 billion of money. They characterize fungal genera, can characterize species, and can even characterize within a species strains. They are biosynthesized by few reaction. Usually are classified in specific group according with their biosynthetic pathways. Chemically are extremely heterogeneous. Can have also some biological activity against plants, not only human and animals, and about the 500 mycotoxin well known, around 10% is considered harmful for humans. Same genus can produce several mycotoxins, but the same mycotoxin can also be produced by species belonging to different genera. The role, well, we can consider, or many scientists consider them as bioweapon for defend substrate from competitors, can be of assistance for being more aggressive toward the host plant, and some other hypothesize the mechanism that the production of mycotoxin for a fungus is the kind of mechanism to maintain metabolic turnover in hard times of the fungus. Or some other have hypothesized a procedure to dispose waste compounds. Natural products that improve the producer's survival fitness, this surely is one of the characteristics of mycotoxin, and they can act by, many of them, by specific DNA receptor in competition with other organisms. So the mycotoxin can be considered as a selected way for the fungi for their evolution, to create genetic variation, and to lead the fungi to adapt better to the different and changing environmental condition. To tell just the importance of mycotoxins, and Antonio later Logrieco will also tell us about the importance of the networking and what is going on with at the European level also. But this is just to evidenciate how the mycotoxin are the most important category notified by the rapid alert system for food and feed of the European Union. This is just a summary of the most important mycotoxin that we will also take in consideration during this talk. We have as main mycotoxins that we work with for Aspergillus flavus, aflatoxin B1. For the Aspergillus niger, clade, and Aspergillus carbonarium, we have fumonisin B2 and ochratoxin A for the niger clade, and ochratoxin A for carbonarius. In Fusarium, we have worked quite a lot on the bovericin eniatins for subglutinans temperatum, which are two pathogens of maize, together with proliferatum and verticilioides, which are pathogens of maize as well, but they produce fumonisins. And another important species on maize is the Fusarium carminianum that produce trichotescens and zarealenone. All these species that I listed with maybe less the Aspergillus carbonarius can be commonly contaminant of maize. The toxic effect of mycotoxins are quite wide. There are some that are carcinogenic, some other hepatotoxic, immunotoxic, nephrotoxic, neurotoxic, and estrogenic. In particular, estrogenic is zaranenone, and in general, there are also some teratogenic, like aflatoxin, ochratoxin, but what is also very interesting that the most important mycotoxins which are regulated by the European Union all have immunotoxicity capability. And here, as you can see, you can find on food, maize in particular, aflatoxins, rarely ochratoxin A, deoxynivalenol, which is a trichotasen, and zearalenone and fumonisin. So the maize is a kind of really collector. Here, just an example about the indication of Aspergillus flavus, which is the main species that produce aflatoxins, is a pathogen that occur on many types of organic material, and of course also in the cereal and mostly on maize. Take a lot of advantage by the hydric stress, water stress of the plant, and it can survive in specialized structure that allow him to survive on the soil for extended periods of time, and we will see how this is important in the last part of my talk. These are the mycotoxins that produce, in particular, aflatoxin B1 and B2, and this is important to this is important to notice for those that are not involved in the mycotoxin study. Is the only mycotoxin that is considered by the International Agency of Research of Cancer Group 1, because it's clear the link between this mycotoxin and the hepatocarcinogenic disease in human. This problem of aflatoxin is something that, according with several studies, and in particular the studies that our group in Bari has participated, led by Professor Battilani from University of Piacenza, we have studied the possibility of contamination of aflatoxin and due to the development of Aspergillus flavus on maize, with a scenario of climate change of +2 and +5, and while for wheat and and rice there is no risk that has been established. If we focus about maize, we have a serious risk of constant contamination in Central and South Europe, with the risk that aflatoxin will be the main problem that we will face with this climatic change ongoing. And in Italy, we are often dealing with this water stress problems on maize, and there are at least in the last 15 years is quite periodical the possibility that the aflatoxin contamination do occur on maize. In addition, we have also, this is a minor problem, but is there are contamination caused also by the black aspergilli, in particular the Niger clade member, that in particular Niger and Wolbiskie. These species can produce on maize the fumonisin B2 because they have this characteristic. What is important to notice is that all species have their own mycotoxicological profile. Therefore, the correct identification is important to define also the risk for consumers and the risk, and so it's important in terms of risk assessment of what you are dealing with. Another problematic for the maize is the Fusarium head blight, and also known as Fusarium earrot. There are species at worldwide level that produce fumonisins like Verticilioides and Proliferatum, which are dominant on maize. Subglutinans temperatum are also dominant, but in different geographical areas, so this can change according with the geographical area. And Fusarium graminearum also, that produce trichotillins, in particular the oxenivalenol, which is an inhibitor of protein synthesis, can occur also when the climate are more cold and humid. In particular, we know exactly two kind of maize earrot, the pink earrot and the red earrot, caused by the fumonisin producing species and by Temperatum and subglutinans, which have a different risk because the Fusarium graminearum, that is the principal agent of red earrot, has a different approach, different biological cycle, while the species that produce fumonisins are carried out by insect as vector, and then is important the insect control as critical point. Fusarium graminearum has not this problem because the cycle he survive on the residues and he attacks the plant at the silk emission. It is also the optimum of temperature and humidity of these two group of different species is different. So, summarizing, we have maize, we have a risk of Fusarium contamination that cause fumonisin contamination, trichotillin producing species contamination, and we have Aspergillus flavus with the risk of aflatoxin, and Aspergillus actionigri with the risk of fumonisin B2. So this leads all scientists dealing with mycotoxin fungi to develop tools for early diagnosis or for easy and identification of the different fungi. Again, because each species has its own approach, and we studied, in particular, our colleague, Dr. Suska, led a group in our institute to develop a single, let's say, kind of barcode for all species that can contaminate the maize. And she has worked on the calmodulin as the work carried out as a focus on this relative distribution K2P distances across all sequence pairs of Aspergillus species for different markers. And she had also work with Fusarium and all Aspergillus and all Penicillium that have been reported in literature as occurring on maize. Has been a huge screening of different species, and she has developed species-specific primers designed in calmodulin gene that have a species specificity by developing species specificity for representative primer pair, and this was assessed with fluorimetric method and real-time PCR. So we could have a bunch of primers built on calmodulin that could guarantee the correct identification among all these species. So most of primer pairs are able to specifically identify single species, and therefore they provide a useful tool for risk assessment. However, the results obtained as well allow us to give an ability for detection of a wide range of toxigenic and phytopathogenic fungi, and this is a starting tool for further and more accurate development. What will be what is the added values of this primer is that they have the potential to be incorporated in a rapid and high throughput screening system, like array cards, and this will allow detection of multiple fungi by simultaneous PCR reaction for each maize samples. This will be a kind of early diagnosis tools, molecular tools to avoid isolation, identification, as you know. There are, of course, when you have different needs, when you need a quick identification. We also work on bovarecin, which is a minor mycotoxin. It causes apoptosis in human cell lines, has no acute toxicity known in animals, but has a chronic effect. Yesterday, I was in a mycotoxin workshop in Tolosa, where colleagues from Norway have proven the toxicity of this mycotoxin together with enietins on salmon, and then because of the feed contaminated by these enietins and bovarecin that in cereals are really spread and occur sometimes at very high level. And so we studied bovarecin in respect to two species, Fusarium subglutinans and temperatum, because originally we identify among fusariologists only subglutinans as able to produce fumonescins. Then there was a reclassification in two that divided the subglutinans in two groups that were based on RFP DNA sequence polymorphisms. These two groups, we wanted to verify if the production of bovarecin was common to the two groups, and actually we identified that only the group 2 was able to produce bovarecin. This is, again, is important because if you have a species that are morphologically identical and they are phylogenetically sister species, it's important to understand which produce what. So more recently, the group 2 has been reclassified as Fusarium temperatum, which is again very closely related to subglutinans because they were identified as group 1 and group 2. So we did a long, big, wide screening to understand if the group 1 was subglutinans first, and then if the group 1 was non-producing bovarecin. And then we confirmed that temperatum strains that we isolate worldwide, this is just a part of a large collection that we have run, only the Fusarium temperatum strains could produce bovarycin. Therefore, is interesting because these two species often co-grow on the same habitat. They are sister species. They just recently diverged. So we wonder why this group produces Fumolicin temperatum produces bovarycin and why Sublutinans do not. So this is just to show how wide was our screening. Then a first approach was to build specific primers, and was done this by designing these primer pairs on the TF, elongation factor alignment. Then was developed a species-specific for the two species, primers which are absolutely able to distinguish the two species, and also to distinguish these two species from other species that occur on maize, always related to Fusarium, like Fusarium proliferatum and verticilioides, that are phylogenetically the most closely related to Fusarium temperatum and Sublutinans. Then we study, by studying two genomes, the gene cluster of bovarycin, and we found the gene cluster exactly in the same position and with the same characteristic of the Fusarium fujicuroi, that was a gene cluster that was identified by Neau's eco-workers. So the entire cluster was found in both species, not only in temperatum, and was complete. There was complete synteny between the two species. In addition, orientation of genes were the same. Then we look here just to confirm, and you can see also the flanking regions that for the group that is also called all these species now are classified in the same Fusarium fujicuroi species complex with according with the new phylogeny definition of the genus. So they had the flanking regions absolutely similar. Then what was studied that there were single mutation in the strains that we look at the genome that could contribute to the non-functional BA1, the non-ribosomal peptide synthase gene. So, and in addition, we found also an insertion in both sublutinans analyzed in the entire genome. So we could found these two SNPs and this intron, and then we studied by developing all the population that we previously analyzed for the production of bovarycin in sublutinans and in temperatum. And you can see that here there are some different combination. There are some group which have the intron and maybe just one of the SNPs. We didn't find never the strains that had all the SNPs and the intron. However, all this group were sublutinans, and they carry at least one of the SNPs or insertion. So our I don't know. It's working.
I don't know. Maybe forward. It's not working.
Okay. No, it's working. Okay. Then what we did is to analyze the the sequences of the BA1, the variation analysis of this BA1, and we look at the variability in temperatum and in sugglutinance, and we observe that in sugglutinance there is a low level of nucleotide variability in the whole BA1, but eventually this causes a premature stop codons and then a framsheet generating not functional enzyme for the bovaricin production. While in Fusarium temperatum there was a higher nucleotide variability, but this didn't cause any biosynthetic enzyme functionality alteration. The conclusion for this part are that species-specific primer were developed and then will allow us to distinguish quickly sugglutinance from temperatum, that the production of bovaricin is only related to temperatum, and that this will allow a more accurate mycotoxin contamination assessment risk, that the entire cluster was located and was completely in syntony with the Fujikuro cluster for both species, and that the single base variability of detected in this BA1 could provide for the non-functional activity of the BA1, that is the most important gene for the production of bovaricin. The nature of this genetic inactivation of BA1 is consistent with its relatively recent occurrence and the close phylogenetic relationship of the sister species. It's important to notice that this improvement in structural variant detection and analysis should be our future priority to better evaluate the impact of this type of variants on evolution. I report here just the example of temperatum and sugglutinance, but we have other example that we have studied of variation intraspecific, where you can look at very population that carry some mutation, like we found in Aspergillus welwischii, that is a fumonisin B2 producer, so is a toxigenic species, where we found differentiation of population that can produce mycotoxin, cannot produce based on some gene missed or some mutation. And this is in the same species. So other example I could do with the Fusarium proliferatum that we are studying from different crops. So there is a very charming field of investigation in on this matter, on the screening biosynthetic pathway genes and the true capability of the genes intraspecific variability of the species to produce mycotoxin. This will be more feasible, of course, with improvement and cost reduction of long-read sequences technology. Finally, just I want to point what we are doing just during this period. I finish. Okay. So I go quickly here. We have some study going on the volatile production by the volatile compounds production by toxigenic and non-toxigenic species of Aspergillus flavus, just showing you that there are in Aspergillus flavus population which are toxigenic and other which are non-toxigenic because they miss genes, and they are proposed as biological control agents for displacement of the toxigenic species. What we wanted to develop in cooperation with the University of Liège, in particular by Laurie Josselin, which is doing this work with our strains, is to understand if there are volatile compounds specific for the toxigenic and for the non-toxigenic. So I really go quickly, saying that we have tested non-toxigenic strain versus toxigenic strain, and one of these toxigenic strains in the lab had a natural variant and lost the production of aflatoxin. From the study, we have evidence that there are 22 VOX common at the 3 strains. So there was alcohol, ester, furan, ketone acid, but in particular 27 VOX that were terpenes were common to all 3 strains. So the problem was if what is the difference distributed among all the families of molecular, or does one of them stand out? So the distribution seems to be equivalent except for the terpene family, in which case 99% is common. Just going to the end, we found actually she found, and this is confirmed by some statistical analysis, that there were a pizonaren that was is a terpene that was only related to the toxigenic strain, while the ethyl-2-methylbutanate was related to non-toxigenic strain. So from the work that she has done, I want just to send this kind of message. We need to go for this kind of approach step by step. So first, analyze the VOX which are common to all fungi, and from literature and from this study, there were some VOX that were identified as common to all fungi. Then try to compare non-toxigenic and toxigenic and see which are common to Aspergillus flavus, toxigenic and toxigenic, and there were some that were common. And then go on the difference between the toxigenic and non-toxigenic, and we at the moment found only a single compound, but the work is still going on, and I think there will be very nice data that are going on, and that will allow to us to have some volatile markers specifically to non-toxigenic. If you think that the non-toxigenic species are biological control, are used as biological control formulate in many areas of the world, to have to screen rapidly if you have dealing with some volatile marker, if you have dealing with non-toxigenic and toxigenic is very important. Also because this is also extremely important, since the non-toxigenic population have a mutation and they miss the genes in different point, to have a molecular marker is really difficult, because you don't know always if you take a gene, one of the biosynthetic gene that is involved in the production, but you don't know if you miss other or not. So for this reason, this way will allow maybe to have more robust marker. Thank you very much for the attention. Sorry if I was long.
Thank you, Antonio. Well, thank you, Antonio Moretti. We'll have to find the time for discussion after, so please go with the next speaker, because we have 15 minutes after. So the next speaker is Antonio Logrieco. He is the director of the Institute of Science of Food Production of the National Research Council of Italy. So please go ahead, and I will take you 5 minutes.
Okay, thank you very much, Mr. Chairman, and thanks for all organizers to invite me in these really interesting events, particularly for the organizer. So my talk is from myco-key to myco-twin, mycotoxin management along food feed chain. So I'd like to give you some overview about these two projects and the different outcomes that we had during this year. So as Antonio showed you, this is the maize is considered in mycotoxin area one of the most important crop risk. So this is together with wheat, of course. And then, so as you can see, so there is different products derived from maize and wheat that can be contaminated by mycotoxin. So just some slides similar to Antonio showed, just quickly. So this is a complex. We have different symptoms. This is pink hair rot and the red hair rot due to a complex of the species, just not one, but this is a complex of Fusarium species occurring on the maize hair rot. And then with the different mycotoxins. So this, we have to take in account that each species may produce different toxins. And then, so there is a complex, in this case, of mycotoxin. Also in Aspergillus genus, there are different species. So we have Aspergillus nigri and, of course, Aspergillus flavus parasiticus. And we have also different complex of mycotoxin, fumonisins, and aflatoxin. So aflatoxin, of course, is an emerging, really emerging, but we can consider already established problem in Europe, starting from 2012 to 2013, when we had a really severe contamination in Italy, Romania, and Serbia. So this is just a map of aflatoxin risk in maize at the global level. And as you can see, so aflatoxin is quite spread, aflatoxin risk, particularly in Europe. And then is really, with the climate change, is coming really one of the most important safety issues. If we move on fumonisins, the situation is almost the same, but of course is more, also in this case, we have a really spread risk at the global level. So this is, I'd like to highlight the importance of the fungi occurring in maize, in the cells, and the multi-mycotoxin that may occur in the same kernels or in the same crops. So this is just an example of how many mycotoxin may occur in these cells in maize. And then this is a slide show you how we can detect also mycotoxin and the same respective species that produce this specific mycotoxin. So this is really a complex situation with a really complex mycotoxin risk. Also, this is due to also the complexity of biodiversity, particularly in Fusarium graminearum. This is just an example, and this is Cheilodonus distinguished 12 different species of Fusarium graminearum, and that due to, of course, of molecular approach. And then also there is a different chemotypes in Fusarium graminearum, as you know, with 3 different toxin production, 15, 3-acetyldeoxynivalenol, and nivalenol. So this is very important to know the spread of these chemotypes, because as you know, each toxins, or each the different trichotices are really different toxicity against the animal and the human issue. So this is, in other words, nivalenol is much more toxigenic than 15 and 3-acetyl. So this map is just as important to know the possible risk at the global level about these trichoticins. This, of course, maize represent one of the most important trade risk exchange, particularly from the all continents, particularly on fumonisins and aflatoxin. And then the regulation of mycotoxin at the global level is increasing every year, because there still exists some country without any regulation. So this is absolutely not acceptable that some country, particularly in Africa, there are no regulation or very limited regulation or different limits of regulation respect of the rest of the world. So this is an unacceptable situation. And also, as you can see in these slides, there are also some significant differences in terms of regulation in food and feed at the global level. And then Europe is more, in food, it's more sensitive, so the regulation more restricted than North America. But if we move in feed, North America is more restricted than, more restricted than Europe. So this is a different regulation aspect in these two important continents. So due to the economical impact and the economical and health impact of mycotoxin, so European Commission dedicated a lot of attention about of this problem, and then so we coordinated the MycoKey project in Horizon 2020, and then recently also we are part of a consortium for MycoTwin, coordinated by Turkey from R18. So it was really a good cooperation between these two projects. In MycoKey, so this is consortium, is a big consortium with the European partnership, is European core partnership, but also with China. And then, so this is was quite effective, this cooperation. And then, so we took in account all the chain from field to fork. And then, as you can see, so the sides of the flag, the Chinese flags, means the interest of the Chinese consortium in the specific action. So as you can see, the pre-harvest was more attractive from Chinese sides. So this is mycotoxin management is quite difficult along chain, and we have a pre-harvest and post-harvest. In the pre-harvest, the situation is quite complex due to the many factors, and the control is, of course, is limited with the problems and the side effects of corrective action. If we move on post-harvest, situation more simple. So this good agricultural practice is limited toxin accumulation is quite effective, and no side effects. So considering different work package, so I just give you some slots, some flash about the outcome of the project. So this is early detection. So we have developed the lamps assay for specific Aspergillus and Fusarium trichothecins. Then also we sequenced different genome of Aspergillus Fusarium species in order to find the biodiversity geographical distribution, and then also identify gene cluster involved in a specialized metabolism of fumaricins and ochratoxin Aspergillus flavus, and fumaricins in Fusarium species. Then this is just a map of distribution of different chemotypes in Europe, and this is quite interesting. And also we extended our study at the global level. So also in this case, we confirm the different geographic distribution of these different chemotypes. So the mycotoxin monitoring. So this is in this case we developed methods. This is very attractive, as just published in Toxins these months. By using the dust and by using LSMS for multi-mycotoxin methods. And this is a very important and cost reductive action in order to predict the possible risk in these in great or big tanks. And then also we developed a multiplex dipstick. We used the aptamer-based for a multiplex strip test for aflatoxin, and new biosensor for platform and biochips. So this is a really important advance with also with the collaboration with China's consortium. So prevention, what we can do to prevent mycotoxin in the field. So this is, as mentioned before by Antonio, so this is a biocontrol of Aspergillus is one of more important and effective action in the field in order to prevent aflatoxin. And then we developed 3 different action. One in Africa, we confirmed in Africa, we extend the use of aflasafe in Ghana with a significant reduction of aflatoxin. we developed this methodology in China and also in Europe, particularly in Romania and in Serbia, with the identification of some Aspergillus flavus atoxygenics, not toxigenic, for using in biocontrol agents with lacking of all genes of toxin cluster. So this is just an example of the number, about of the huge number that we studied in order to identify the atoxygenic strains. We conferred also by molecular approach. And then so we studied different fields. We used these agents in order to control aflatoxin in the field in Romania. Breeding, this is your field. So we identify different maize line resistance for Fusarium earrot, and then so against Fusarium verticilioides and Graminearum with the specific molecular market and with the linked resistance in Italy. But also we extend also on wheat. But it's interesting that in China they really studied deeply this aspect, and they use a different maize germplasm from different parts of the world. China use Canada, Romania, Germany, France, Netherlands, and so they found a good resistance against verticilioides with 11 lines resistant to Fusarium graminearum, and then 33 lines resistant to Fusarium verticilioides. This is really, I suppose this is very interesting outcomes. And interestingly also this study was conducted with Lanubile. So this is, I'd like to point out that this infection with Fusarium verticilioides give the induction of about over 80 early genes and 24 late genes. But the interestingly part of this study was that in resistant maize, most of the gene provide a basis level of defense to the fungus prior of the infection. So this is the resistance was very reactive to the infection. The susceptible maize, most of the PR genes are induced after infection. So that means it's very, very important way in order to possible link this reaction with the resistance of maize against some Fusarium verticilioides. So another activity was pre-harvest strategies by using isothiocyanate, this from mustard based botanical, and then so we used these volatile compounds in order to avoid to reduce the contamination in the silo. So with this in lab scale and also in real scale. So this was very effective, and these slides show you the effectiveness of this methodology in order to reduce contamination. Interventive strategies, so this is 80%, so this is just reduction of disease severity of the ON by also using chlorostachics or Trichoderma arthrobrunneum item application in the soil in order to reduce the inoculum of these bactoxygen fungi in the soil. Remediation, so this is a different approach, but I'd like just to highlight this, the use of mushroom in order to reduce, minimize mycotoxin, because these mushrooms, Pleurotus, produce enzyme, particularly lactase, that is able to degrade many mycotoxin. And in particular, so this is just an example of how this is the mushroom that's growing on contaminated substrates, and then so the carpophory grow very well without any contamination, and then we obtain the degradation up to 86% of aflatoxin B1. Interestingly, that we got has a production. A degraded production aflatoxylol was found mature, no sorry, no aflatoxin B1, neither aflatoxylol was found in carpophoris. So that means that the Basidio carpus was quite safe. So we used the lacase in order to get the combination lacase expression in Saccharomyces cerevisiae in order to produce lacase in huge amounts. And then we used this enzyme for degradation of different toxin. And then, so in real samples, we obtained a reduction of 40% of zeranone in maize and aflatoxin M1 100% in milk. So this is quite really interesting possibility. So as a product, so we obtained aflatoxin Q1 that is 18 times less toxic and non-carcinogenic by HAMS than aflatoxin B1. So then also we used these Saccharomyces cerevisiae with the gene involved in lacase production in order to use in the digester to obtain the biogas. So this is quite useful possibility by using, of course, contaminated batch, because the regulation do not allow to use the contaminated batch for animal feed or other purposes. So in this case, we can use it. So this is just some paper. So this is also ICT solution by using a different approach. This is the microkey hub in order to predict possible contamination in the field. And then, so by using also this drone approach. So what I'd like to, just these 5 minutes, I'd like to highlight the outcome of organization of nominal groups. So at the end of the project, we organize the different groups, and so we discuss about possibility to manage different mycotoxin in pre-harvest. For example, in this case, we have as a first option that we obtain is the genotype selection for Fumonis management. Then we did 33%. Then we had the insect management, 23%. That means these two factors are very strategic, very crucial in order to manage Fumonisins in maize. If we move for example, in aflatoxins, the situation is more or less similar, but with some significant differences. Genotype is still the more considered action in terms of management, but then 23% is also the biological control. So this is it can be considered the biological control one of the also together with genotype selection one of the most important action. And then 50% is water management. Then if we move on the oxymanol and the zeranone, genotype selection is the most important action. So with the 36%. And then followed by crop rotation, 14%, and insect management for 9%. So priority for decontamination, detoxification of mycotoxin contaminated maize grains. We have a different option, different possibility with the optical sorting, 18%, removal of fines, 17%, then density separation, 14%, milling 30%, chemical detoxification 11%, adsorbents 10%. So this is an interesting possibility. Then for post-harvest management mycotoxin, we have a rapid drying 18%, 15% cleaning, sorting grains, 30% harvesting, handling equip, 12% good storage facilities, 12% harvest timing, and 10% is humidity and tempered in storage. So it is very important to control these two parameters. And finally, priority to new research information that will most effectively contribute to improvement mycotoxin management or reduction in risk. We have 24% as a risk assessment tools. 70% is plant breeding technology, so this is still one of the main crucial action. And 50% is management guidelines, and 11% is information transfer, and 10% is technology adoption. Okay, so just the fine conclusion. So this microkey was awarded by European Commission for a success story from EU research in order to facilitate management of mycotoxin in a long food chain, and also to improve the cooperation between EU and China at that time. So we are really great human relationship for growing international network. This is a different moments, different location that we had during this project. And I'd like to thank you for your kind attention.
Well, thank you very much for being so punctual, and we are on time. Maybe we have to connect with Ras Bernardo now, and if he is ready, and then we have the discussion after. We are just on time, so. So next speaker is Rex Bernardo, is professor in corn breeding and genetics at the University of Minnesota. Unfortunately, he cannot be here, so we had to connect with him by. Okay, hello, thank you for coming or for staying with us. Do you hear me or?
Yes, I can hear you. Can you hear me?
Yeah, perfect. Thank you. So you can begin whenever you are.
All right. Well, thank you very much. Good afternoon to everybody. I trust and I'm sure that the conference is going well. I'm very sorry that I'm not able to be with you in person today, but I'm glad to at least be able to join you via Zoom and to present some of the results of my work. The title of my talk today is, Should Maize Breeders Routinely Do Recurrent Selection? And I thank the organizing committee for accepting my one-word abstract. If you look at my abstract, there's one word, and the answer is yes. Yes, I think maize breeders should routinely do recurrent selection. And what I would like to do today is show you, in my opinion, why maize breeders should routinely do recurrent selection, even in the context of line development in a commercial breeding program. So I'm going to go ahead and share my screen. All right, can you see my screen well? Okay, so if you go back to the year, say 2010, and look at how maize breeding was done, it probably was something similar to this. It was mainly phenotypic selection with field trials. So a breeder would choose two parents to cross to form an F1. From this F1 cross, say 150 or so doubled haploid lines would be developed. And then these doubled haploid lines would be test crossed to a tester, one or two testers, initially just one. These test crosses would be evaluated in yield trials, and the best would be selected. So for our purposes today, I'm keeping the number of selected constant as 10. And when I say yield trials and evaluating doubled haploids, the understanding is that that is always in test cross combination, not doubled haploids per se, but doubled haploids crossed to a tester, and they are evaluated in yield trials. Now, if we look at the current costs of phenotyping, I think at least in the United States, we could assume that we have a constant cost of $14 for a yield trial in one location. Okay, so we could grow a test cross in a yield trial at one location, and that would cost us about $14. We could assume the same cost for developing a doubled haploid line, meaning crossing to a haploid inducer and then finding haploids and then doubling the chromosomes by colchicine or another treatment. Let's say that costs about $14. And we can also assume that the cost of test crossing to cross one doubled haploid line to one tester is equal to 14. Might not be exactly the same, but the equal cost it's close enough, and it makes it the comparisons straightforward. And then if we look at the main trait of importance in in maize, which is grain yield. The heritability obviously varies, but on average, heritability in a large set of biparental crosses that we have studied is about 0.6, meaning 60% of the observed variation is due to genetics and 40% is due to non-genetic effects. And so if we assume these costs, and if we assume that on average 150 doubled haploid lines are test crossed and evaluated in yield trials at 6 locations, then the cost of breeding with one biparental cross comes to $16,800. And the way I look at that is the cost of a new, inexpensive, and a new cheap car. And if I look back in the 1990s, and it was the same, I thought, oh, the cost of breeding with one biparental population is the cost of one cheap car in the United States. So that is the cost. And if we look at the time component, let us assume that the F1 cross is developed by November 15 in year 1. November 15 in year 1. Now, there is no way that the yield trial can be conducted in year 2. There is no way that between November 1 and the time that the plants have to be planted in year 2, that doubled haploids can be created, doubled haploids can be induced, and they can be test crossed to a tester. And so therefore, the yield trials here are conducted actually by year 3, and we're going to assume that the seeds need to be available by March 31 in year 3. And so what this gives us then is a 500-day window to do different things in the breeding process. And so there are 500 days between F1 and yield trials, and we have a budget of $16,800. And so the question that I was looking into is, if we assume that the time remains constant and the amount of money stays at $16,800, can we do a better job in breeding using the new technologies that we have? And that technology would mainly be genome-wide prediction. And so the cost then is assumed to be equal to also $14 for SNP genotyping. So we're assuming that the cost of phenotyping is the same as the cost of genotyping for several hundred SNPs. And now we're looking at a range of predictive abilities. Prediction accuracy, which is the correlation between the marker predicted performance and the genotypic value. I considered a range of 0.2, 0.4, 0.6, and 0.8. In a way, it did not matter. It doesn't really matter how we arrive at these prediction accuracies. The assumption is that we have a calibration model that leads to these different prediction accuracies. So with that as the background, the question is, what else can we do to try to increase gain from selection? And one approach that already has been used by breeders is what I would call as one cycle genome-wide selection. In one cycle means that, well, we apply SNP genotyping at one point in the breeding process. We use genome-wide prediction. We select the best. So that's genome-wide selection. And then phenotypic selection occurs, or it takes place, for the candidates that have passed the genome-wide prediction process. So we could do one cycle genome-wide selection, still create doubled haploids from the F1, but now these doubled haploids are genotyped for SNP markers, several hundred, at the cost of $14 per sample. The best doubled haploid lines are selected. And again, the prediction accuracy differs from 0.2 to 0.8. These selected doubled haploid lines, far fewer than, are evaluated in yield trials, and the best 10 are selected. And then we can compare what is the gain from having the best 10 from phenotypic selection and the best 10 from genome-wide selection. And this is what I've found. I've found that with one cycle genome-wide prediction or genome-wide selection, the gain is greater than the gain from phenotypic selection by 110 to 132%. Again, to say, we keep the same cost, we keep the same time. We now add one step of genome-wide prediction. We select the best doubled haploid lines. Only the best doubled haploid lines are put in yield trials. If we do that, then we can increase gain by 10 to 100 to 32%. The 10 would be from the lower prediction accuracy. The 32% would be the higher prediction accuracy. So from this, then, it means to say that there really is no reason to not use molecular markers in genome-wide prediction, because we keep the same cost, we keep the same time, and we get more gain in the end. But it seems then that that is not the best that we can do, because there is a 500-day window in which we can do different things. Before I go into that, though, I also studied two variants of this one-cycle genome-wide selection process. And this first variant involves creating F2 plants and inducing doubled haploids from the F2 plants. But we could also apply genome-wide prediction among the F2 plants and then induce doubled haploids only among the best F2 plants. And then we have the doubled haploids, we apply another round of genome-wide prediction, and we proceed as with the first case. Or a second variant is that the genome-wide prediction is actually before doubled haploid induction. What I found is that there is no difference between these three schemes. There's no difference between the main scheme and these two variant schemes. So again, so far we're saying one-cycle genome-wide selection is superior to phenotypic selection. But what if we can do 2 cycles of genome-wide prediction and still make it fit in this 500-day window and keep the cost the same? Can we do that? And the answer is yes. And this scheme then is shown in this diagram over here. So we start as usual with the F1 cross that is identified by November 15 of year 1. From the F1, we get the F2 generation. We genotype the F2 plants, and then we select the best F2 plants, and then we intermate the best 8 to 10 F2 plants to form cycle 1. Then from the cycle 1, we induce doubled haploids. We genotype the doubled haploids with SNP markers. We then select the best doubled haploids, and these selected doubled haploids are then subjected to yield trials. So we see here that there is still phenotypic selection in the end. There still is genome-wide prediction among doubled haploids, and the best doubled haploids are selected and subjected to yield trials. But the key difference is there is one step of genome-wide selection among F2 plants, and these best F2 plants are intermated to form cycle 1, and this is a key step. We are then leveraging or taking advantage of the power of recombination to shuffle the genes which are now at a higher frequency because of this prior genome-wide prediction. And what I found is that if we look at the gains from this process, the gains from 2 cycles, of genome-wide prediction or genome selection are greater than the gains from 1 cycle by 112 to 135%. Okay, so this generation of selection of and recombination again leads to an extra gain over the 1 cycle, but compared to phenotypic selection, we have a gain of 124 to 178% depending on the predictive ability. So the point being is that with 2 cycles of genome-wide selection compared to either 1 cycle or phenotypic selection, we do get an increase in the selection response. And so and remember that this is, again, it's a bit tight, but in an article that that's been published in Crop Science, I indicated these different steps and how they can fit in this 500-day window, especially if we use year-round nurseries, if we use embryo rescue and other ways to decrease the generation interval. All these steps can fit within this 500 window, so we're not increasing the time required in breeding, and again, we are keeping the same costs in breeding. Now, if we look at what resource allocations are the best for this two-cycle genome-wide selection, the following lead to the largest gain. It would involve 200 to 240 cycle 0 F2 plants, okay? Meaning that at this stage, at the F2 stage, we need to generate about 200 to 240 cycle 0 F2 plants. We then select the best 8 to 10, and then we generate, again, about 200 to 240 cycle 1 S0 plants, which now lead to doubled haploids. So we still generate a large number of doubled haploids, 240. We then use genotype these lines with markers. We select the best 35 to 45 doubled haploids, which go into yield trials at 12 locations. And so what we're seeing here is that we are, in essence, skipping, sorry that my screen is not showing there, sorry. What we're seeing here is that we are essentially skipping what is typically called a stage 1 testing. In maize breeding programs in the private sector, there has been called stage 1 testing, which is conducted at say 6 to 8 locations. And then the stage 2 testing, which of the candidates which pass stage 1, that's a larger number of locations, around 12. So what we're seeing here is that we are essentially substituting genome-wide predictions for this first stage of selection. And then those candidates that pass the genome-wide selection process are then evaluated immediately at a large number of locations, at 35 to 45 locations. And so you may ask the question though, well, I mean, is that really, is that the limitation of phenotypic selection? And I've been working on this idea of, or this topic of outliers in breeding. And if we look at different species, including maize, there have been different outlier type hybrids that have extremely good performance, way above the best. And I was interested in the question, well, are outliers rare only because the population sizes that we use are too small, or are they truly unpredictable? And so what I did is I used a different genetic model for barley this time, but it's still comparable to maize because the gains are expressed in genetic standard deviation units, and in both cases, a large number of QTL was assumed. And so if we look at what is the distribution of outliers in a breeding program, this is the distribution of outliers in the top 1%. So what I did was I simulated in each cross 10 million lines, okay? Nobody obviously would create a population with 10 million lines, but it's just simulation, so I could simulate a large number. So I simulated 10 million lines in each of 105 biparental crosses for a total number of lines of 1.05 billion doubled haploids, and asked the question, what is the distribution of the performance of the top 1%, which is the top 100,000 lines, and then the top 0.1%, and then the top 0.1%. And we see here that if we look at the distribution of the top 1% of lines among the 10 million lines, we do have a continuous distribution. It still actually is a bell-shaped curve. Now, if we look further and look at the cutoff, this now is the 0.1%, okay? And we look at the distribution of this 0.1% in this next graph, we see that we still have a continuous distribution. And furthermore, if we now look at the top point, 0.01% on this side, we see that even in the top 0.01%, we have a continuous distribution. So the lesson here is that outliers in a breeding population behave in a predictable manner, but they seem unpredictable only because the population sizes that we use are nowhere near 10 million lines. We're talking more of 100 lines or so that we typically have in a breeding program. Now, if we look at what is the maximum gain that we can get from selecting the top 0.01% of 10 million lines in the population, the maximum gain that we can get is 3.32 standard deviation units, okay? We can get a gain of 3.32 standard deviation units if we select the best 0.1% of the top 10 million lines. However, this is assuming a heritability of 1. Now, if we now have a heritability of 0.6, then this gain now corresponds to roughly 2 standard deviation units. So again, very stringent selection in a very, very large population, the gain that we can get at most is 2 standard deviation units in terms of genetic gain. Now, for the 2-cycle genome-wide selection scheme that I described, 2-cycle genome-wide selection scheme with, again, heritability of 0.6, the maximum gain was 2.13 genetic standard deviation units. And we see here then that the gain of 2.13 with far smaller populations is larger than the gain that we would get if we grew 10 million lines, which means to say then that as conclusions from this study, that there is a lot of power in doing a form of recurrent selection. So in the current timeline for a breeding program, right now we can do only 1 cycle of recombination, selection and recombination. It obviously can be more cycles if we are wanting or willing to extend the amount of time that is required. But if we keep the same time requirement, year 1 to year 3, and if we keep the same budget, there is a lot to be gained from 2 cycles of genome-wide selection, followed by phenotypic selection. And that is why, in my conclusion, I say that yes, maize breeders should routinely do recurrent selection. Thank you so much for your attention today. Thank you.
Thank you. And thank you also for saving so much time. We have 25 minutes for discussion. Is there any question in the public before I begin? So I have the first question for probably for Antonio Logrieco. I don't know, but when you study the risk of mycotoxin contamination, do you consider different genetic system, like for example, organic agriculture or low input agriculture? So, because I saw that you consider actually irrigation, fertilization, and all those things, so that's perfect.
Yeah, of course, there are many actions in order to minimize the possible contamination. No? So this is what we did is also the internal discussion in different groups, is nominal groups. And then, so there is depends of which mycotoxin we would like to minimize. We like to prevent. It has many different actions in order to prevent, also to prevent in order to prevention of mycotoxin accumulation in the field. But once there is already some contamination, we have to also to have some possible handling or suggestion to reduce the contamination for use of food and feed using. So the agronomic issue is very, very important. Is one of strategic action that we can use of course, in the field. And then it consists of course, the irrigation, insect control, biocontrol agents. All this stuff can, of course, induce to get healthy plants. And healthy plants means also more reaction against pathogens of mycotoxin fungi. So this is.
Thank you.
You're welcome.
Does anyone have a question? Yeah. Okay.
I have a question for Rex Bernardo, if he's still there behind the screen. And my question is, how realistic is it for the cost of those three components to be $14? What difference would it make if there was a large variation amongst those components in those unit costs?
Thank you for that question. I think the costs are fairly accurate. I mean, if you look at third-party service providers for phenotyping in the U.S., they would tell you that the costs of phenotyping are around $13 to $14. Of course, it would differ with larger scales. For genotyping, it's hard to keep the costs, but I think it's quite close to around 14 right now. If you look at the cost of test crossing, it might be actually a bit less. And so I think the results are quite robust with regards to costs. What would happen is that if the costs vary a lot, what would differ would be the allocations of resources, how many F2 plants to evaluate, how many double haploid lines to create. But I think the fact that the power of recombination does not change, that power of recombination in selecting the best and intermating them to get the next cycle of selection, that would not be affected by variations in the costs of SNPs and versus phenotyping.
Okay, so Rex, probably you cannot see me, it's Alain Charcosset.
Hello, Alain.
So,
I could recognize your voice.
So, thank you very much for this talk. I must say that besides being innovative in our field of research, you're also innovative in the way of presenting, and from what I heard from the audience, it was very much appreciated. So that was really a nice talk. My question, I think you made it very convincing. The only thing is that if you push the system at the extreme, you will end up phenotyping only outliers that are extremely good. So in the end, you may expect being mostly with extremely good materials. So probably to train the model to be predictive, you need to come to some of your other ideas and to have a different set. I mean, the trials cannot by themselves be probably a good way to train the model, and so probably you need to devote some other means to train the model. Can you comment on that?
That is an excellent point, because the individuals that are phenotyped, they form a potential training population or populations for future populations. So yes, that is an excellent point. Some work that a former PhD student and I have done actually addresses that point. And what we have found is that in addition to selecting the best, you need to select a few of the poorest lines. And we recommended 5 of the poorest lines. So maybe you'll select the best 40 or so, best 40. And then you also need to select the poorest 5, knowing that the poorest 5 should not give you anything good, but it is an investment for the future effectivity of genomoid prediction. And so yes, we found that selecting the poorest 5 maintains the accuracy in future predictions, but it also is a nice check for the effectiveness of the process, meaning that if I now go to my phenotyping and I found that the poorest 5 predicted to be the poorest are not any different from those that are the best, then I know that that there was something wrong, and that the genomoid prediction did not go as well as I wanted.
Okay, so, well, thank you very much. So your rough estimation is that 5 versus something like 40 would be appropriate proportions, right?
I would think so. I mean, breeders obviously would be hesitant to put too many. The difficulty is that now when we're starting with multiple trait selection, that the poorest 5 for yield might not be the poorest 5 for moisture. And so then that becomes a bit more tricky that way. But selecting a few poorest 5, I don't know exactly what number if you have multiple traits, but that seems to be a good solution.
Okay, thank you very much.
And this mode of presentation must remind you of the old days when we had transparencies and we could write on with colored pen. So it's a fun thing to do.
Please go ahead.
Yeah, hello, this is Thomas here. I also have, first of all, thanks for the very motivating talk. I have a question relating to this multi-trait selection. So I guess your assumptions or your results are based on yield only. And if yes, but breeders have to consider multiple traits in their selection in the end. So what would you recommend how to do this? Include this already in the first step or in the genomic prediction, or in the end in the phenotypic selection?
I think that the selection process would proceed in the same way, that you would have predictions for the important traits. So even among F2 plants, you could predict yield, moisture, you could predict height, you could predict flowering date, and that you would select for multiple traits in exactly the same way that the breeder has been selecting before. I think obviously when we have selection for multiple traits, then the gain from each trait will be less than if selection was for that trait alone. But the lesser gains for multiple trait selection, you would see that regardless of the breeding method, whether it be phenotypic selection or 1 cycle or 2 cycle genome-wide prediction.
So is there any other question? Oh, Alain again.
Thank you very much. It's more a question for the two colleagues who spoke about mycotoxin. So I don't know so much. Could you comment on your views on what has been achieved in terms of genetics? Did things change a lot? And is there a long way still to go? Can you give us? I saw that 33% for some aspects were from genetics. Where are we now, and where should we go as a community in terms of maize breeding for these traits? Can you comment on that, please?
Please. Do you mind if you can hear? Please, sorry for the inconvenience.
Well, there is Antonio showed something about some resistance lines for fumonisin and aflatoxins that were achieved by using molecular markers, and they are really promising. So it's a good direction, and I do think this is a very promising achievement. In durum wheat, for example, we have for the fusariosis still a lot of problem due to the fact that when you can select varieties which are more tolerant or resistant, then you lose productivity and quality. And that is the key point for the durum wheat, for example, which still has a very poor resistance against the fusarium mycotoxin, the mycotoxin produced by fusarium. So it depends from crop to crop. Seems that on maize, for what is related to fumonisins, they were the group that cooperate in MycoKey in the project that has explained as shown Antonio were really also excellent in terms of combination with the productivity and quality of maize. On durum wheat, we are still there is still a lot to do.
Jose.
Hi. Hi, Rex. This is Jose Crosa. Thank you for the talk. I wanted to comment that perhaps on the 5 worst lines or predictors, you might use a selection index if you have a multi-trade. So you might select the 5 or the 10 worst entries based on a selection index. And that will solve the problem of the multi-trade that you were talking about.
Yes, I would agree with that. But again, depending on the traits, for example, if you use a multi-trait selection index, and if you're selecting for height, then, if you're selecting for yield, moisture, and then height, if the weights are strongest best for yield and moisture, then the lines, the 5 poorest lines might have lowest yield and moisture, but they might not have the poorest plant height. And so, I think it would help solve that problem. But I think a key is that you capture the tails. You're selecting the best, but you're capturing also the other tails so that you can maintain that prediction. And so, really, I think what needs to happen is some way that you could effectively capture the lower tail of that population, and whether that is using an index or using an index for some of the entries, but then adding a few more that are low for that are too tall if you're selecting for shorter plants. So I think it might have to come from a variety of approaches, but the main point is that if you select for the best and have a few that are poor, then that seems to maintain the prediction accuracy, future prediction accuracy.
Just to add something about your question. In some cases, we got a really interesting results in terms of resistance. So once some lines show some resistance against, for example, Aspergillus, these same lines show also some resistance against Fusarium. So probably these the same resistance genes can act against the different plants. And then I'd like also to discuss about the use of also the new technology in order to predict all these new technology to predict some markers, specific markers, in order to find out about resistance in some susceptible resistant lines. So this is very useful and predictable methodology. So that I think that could be very useful for all of you in terms of breeding, in terms of find out the possible resistance line. And then one more thing that I'd like just to put on the table is the use of OGM, of course. This is a very important issues that we have to take in account in order to reduce and minimize mycotoxin issues. For example, as you know, the BT corn is quite resistant against the insect, and at the same time we have also the consequence in order to reduce fumonigens, because this connection of Fusarium verticilloides and the insect attack against plants, or some genome other technology that is now using. So this, I think that there are many technology that can be adopted now in order to speed the possibility to up the resistance in a different line.
Any other question now? Yeah, Elliot again.
Thank you. It's again for Rex. If I got it right, in the scheme you showed, you start from an F2, and then more or less you go very deep in the genetic gain in that F2, and then you recycle. Do you have some guess of what would happen if you would recycle between F2s before that?
That's a great question. No, I did not look into that, mainly because I think I don't really know how much breeders do that sort of thing. I think breeders are very much focused on looking at one biparental cross at a time, and as you said, going deep into that biparental cross. But I think that's a great question. If you select the best F2s and you begin intermating with other populations, that's a bit difficult to study via simulation because it opens up a lot of possibilities. And so I haven't done that, but that's a great point to consider. I mean, there's certainly, and it's not one or, I mean, not necessarily one or the other. That it's a possibility that both types of procedures can be done. But yes, a great point.
Okay, thank you very much.
So more questions? Well, meanwhile, I have a question for Rex Bernardo. When we do, normally breeders that work with germplants, we work with local populations that have deleterious alleles, problems of adaptation, and health problems, and anything like that. So I don't see how to quantify those things, and most times we just carry out subjective selection based on scales or general impression of the plant. Is I suppose when you are talking about recurrent selection, is just focusing on the classical recurrent selection that conduce to superior hybrids for yield or something. Am I wrong? Or
No, I absolutely agree. And so the context of this work is, it's more in the context of a company breeding program, of how in a company breeding program, the workflow can be organized, that the procedures for creating double haploid lines, the logistics for genotyping, that they all work together. So the context certainly is in terms of elite material in a company breeding program. Now, if you're talking about breeding with less elite material, then there certainly are other issues to deal with, lodging, flowering date, and so forth, that need to be cleaned up or addressed before you can apply, I think, a very aggressive breeding program, such as the one that I have described.
thank you. So more questions? We still have time for discussion. I also have another question for mycotoxins, as nobody else is. Sorry, there's a question there.
I have a question for the mycotoxins. How durable is durable resistance in maize? If you start breeding for a specific gene, by the time that becomes a new variety, has the disease evolved?
Well, this is a crucial question because, as many of you know, the Fusarium and the toxigenic fungi have a great capability to mutate and to escape tolerance resistance. Regarding the last achievement of maize, still there is time because they are going to let's say have a wide production of these lines. It's not they are really recent achievement. Actually, we have also to, as Antonio said, there are some kind of fungi which against which you don't. You cannot use the resistance line because they are more aggressive through the vector that are the insects. Then you can have very excellent lines that escape the contamination, the inoculation, the attack, the colonization, but then close to the maturity of the harvest, there are these insects that arrive and then can carry over the spores, and then you have a dramatic attack. For this was proved by colleagues that the BT corn was much effective against the fumonisin contamination. Against deoxynivalenol and other, or Aspergillus flavus, there are more data. However, still the new achievement don't allow us to say anything about how stable will be this resistance. On the other hand, on Fusarium, on the Fusarium on wheat, on durum wheat especially, or also on soft wheat, the let's say, is a continuous fight, because you have good lines and after a while you have new varieties of pathogen that can escape. So it's something that cannot always let you stay on your, sit on your success, but you need always to, the Fusarium especially are pathogens which can mutate with a great capability of adapt to new conditions, new parameters, and new then also resistance lines. I don't know if I reply.
So we might have time for a short question, if, or we can keep. Sorry, who? I don't know. What? Ah, the chat. I don't know. Can you see the chat? Okay, good. So maybe we can. No other questions? I just a last one. So when you were working on, you were talking about resistance of some on maize to Fusarium contamination or to mycotoxins contamination. I don't know if you have ever considered other special types of maize, like sweet corn, popcorn, or the different endosperms. You probably haven't gone so far in this research, right?
Yeah, we are, frankly, so I am not breeders, so I am microbiologist. So we have a different collaboration, but the main goal is to the lines not for food consumption, but for feed consumption. So this is for white. So this is for production for feed consumption. So one more thing that I'd like to considering the resistance is that is not, of course, is not simple to handle these issues, because depends of fungi by fungi, genus by genus, species by species. But there is also some interesting factor that we have to take into account. That is, for example, for aflatoxins, there are some asymptomatic kernels without any symptoms that can be contaminated by aflatoxins, and then also contaminated by Aspergillus, but without any symptoms. That means that sometimes Aspergillus flavus may occur also some like saprophytes or insides of kernels. So this is quite complex, the situation. Of course, there are also other factors like insect or other parameters that can induce, of course, this attack or this contamination, colonization. So this depends of species by species. As I showed you before, so the more related, the more important aspect that the breeders can act is against Fusarium graminearum, that is real pathogens. This is these pathogens doesn't needs any other factors like insect or other, and sometimes also is Fusarium verticilloides also is important as a saprophytes follow the plants without any symptoms too, and then when the plants is in not good shape, then colonize and then produce huge amounts of humanity. So that this is not easy to. But anyway, so we are here just to find the solution, to find the resistance.
Okay, so we are just on time. Thank you very much to the three outstanding conferences we had today and to the nice discussion. So thank you. And now it's time for a sponsor presentation with Winterstegen. Do you want to sit down or do you prefer to? Okay.
All right. Okay. So, hello everybody. First of all, thanks for having me. My name is Max Meyer and I work for the company Wintersteiger, which most of you probably know for sowing machines and harvesters. However, and I hope I'm not going to disappoint too many of you now, I'm not going to talk about harvesters or sowing machines today. But I would like to show you a little bit what Wintersteiger has in store in terms of digital solutions in plant breeding. And in particular, I would like to talk about, here, that's how it works. I would like to talk about...
No, this is the pointer. This is to go forward.
Okay.
The big one, no, the big one.
The big one is the pointer?
No, the big one is the... No. The big one is to go forward.
Yeah, okay, but the pointer is the one up here, right? Yeah, it was just... Well, I would like to talk about, in particular, drones and drone picture analysis and about our plant breeding software, EasyBreed. And I would like to start with drones. So for the drone part, we are cooperating with two French companies, which are Delair, which is more on the hardware side. So Delair is a manufacturer of drones. And on the software side, we're working with a company called Altea. So this is the software that you need to analyze the pictures which you have taken with the drone. And as you can see, the drone that we have in our portfolio is a fixed wing drone. And more importantly, maybe, the camera that is built into this drone is a MicaSense Red Edge MX. And this camera is a multispectral one, and it has, it collects data in 5 different bands, which is red, green, blue, so normal digital camera. Then we have one for the transition between visual red light and the infrared light, which is the Red Edge, and one channel for near infrared light. So just for you to see how this drone is operated, it's actually pretty easy. You only need a tablet, and you have a software on this tablet. And in this program, you tell the program the area you want to fly over and take pictures of, and you tell the program where the wind is coming from, where you want to land, and where you want to start. And then, as you can see in the picture, as you can see in the picture, the software will automatically create a flight path of the drone, and all you have to do afterwards is you click start in the software, throw the drone into the air, and the drone will automatically fly above the area that you have selected, take the pictures, and then land. Then we have taken a lot of pictures, but we don't really get any information just from looking at those pictures, right? So we need to analyze those pictures, and that's why we are working together with the company Altea. It's a web-based platform where you upload your pictures. However, I want to mention here, Altea can operate with any kind of drone and any kind of sensor, and also any kind of drone sensor combination. You're not restricted to the drone that I've just shown you. However, they work pretty well together because they're set together. But you can use any other drone on this and any other sensor on this platform as well if you like to. So the first step Altea does then is the so-called photogrammetry. So we're stitching together the individual pictures that the drone has taken into one big one. And the big picture is then called auto-mosaic. And then we can insert our plot grid, we can insert our plot grid in this big picture, and we can check for certain traits of our plots, which is first plant height, which is possible for any crop. Then we can look at plants and gaps in the plot, so we can count the plants and the gaps. However, this only works for row crops. So maize, as you are all concerned with, works, It's not yet possible for cereals. However, I know that Altea is working on it. They might publish an algorithm soon and have it in the software to also be able to count cereal heads. Another thing that we can do in all crops is fraction of vegetation cover, which is basically soil cover. Then we have one trait, which is the flowering time. However, this can only be measured for crops that flower yellow, so canola and sunflowers. And the next trait is then again possible for all crops, emergence. So in an early stage, you can fly over it and see which genotypes are emerging better and which are emerging worse. And as a last trait, we can calculate the so-called stay green index. Some people have talked about that already here, where you can basically distinguish if genotypes late in the season senesce early or later. And then, of course, you can calculate a lot of different vegetation indexes. I think the most prominent one is the NDVI. Everybody knows that one. And you can also plot-based get the results of these indexes. Also, if you're not satisfied with the indexes that are already in the software, you're always free to type in your own index formula, and the platform will include that and calculate this special vegetation index for you that you want to have. And here, just for you to see what the software looks like. So this is a maize trial that we did at Wintersteig in Austria last year, and you can see this is the plant height. And you can click on individual plots. As you can see here, this plot is clicked on, and then it will tell you the mean height, which in this case is 2.3 meters. And of course, you can also extract all of this as CSV files and then convert it into Excel and use it in your breeding program. So now we've generated a lot of data with drones, and not only drones generate a lot of data, but also harvesters, as you know, and probably mostly lab or genomic data is quite a lot. And conventional data management solutions like Excel, they're getting to the limits. And that's why we have at Wintersteiger a plant breeding software which is called EasyBreed. And our vision for EasyBreed is to be able to communicate with as much devices that generate data as possible. So we want to be able to give outputs to machines that generate data and then import the data that they have generated directly into EasyBreed again. And this not only for Wintersteiger machines, we want to communicate with all machines from other companies as well, so you're not restricted to the Wintersteiger family. And yeah, well, as you can see on this slide, we're talking about planning, GIS-based planning, then generating outputs for sowing machines, field plans. Later on, then if you're scoring, we talked about drone data, and of course we are able to generate outputs for harvesters and then take in the data that they have collected. And in the end, you can run your analysis with R scripts directly in EasyBreed. And a lot of things are done in EasyBreed based on R. So for example, if you have a special statistic analysis that you want to use, we can always integrate your scripts into the software, and you can continue using the same analysis that you've used before and that you're used to and that you need. But EasyBreed is not only a data collecting device, it's actually a software that supports the entire plant breeding process. So we have tools for crossing, where you can generate a parent list and then do your crosses in the software, manage afterwards your pedigrees. Then when we're going into trials, we have tools for trial designs where you can do randomizations. Again, this is based on R. If you have a special randomization, we can import it and you can use it in the software. Then we can, if we're going to sowing, we can create field plans for the sowers or for yourself for orientation on the field. We can then assign seed lots to different trials so that you always have an overview if you have enough seed sources for the trials that you want to plant. And then, of course, we can generate working lists, which you can take into the lab and then fill in your seeds into the sowing bags. Yeah, then of course we can create field books, like either to print out and write in manually. We can communicate with certain apps like the field book app, or as you've seen before with the speech recognition software Smetrix. And then when we look at the integration of data, I think I've talked enough about integrating of data, but it's the core of the software. And afterwards, you can then analyze your data. I've also talked about that with your own statistical analysis that you're used to. And in the end, the most important step probably, we have a tool for selection, so you can select the most promising genotypes and then move on with them into the next year. So just for you to see what the software looks like, we're working a lot with dialogues. So here on the left-hand side, you can see a dialogue to create a field design. First, you would choose which kind of randomization you want to have. Then you tell the program which experiment you're actually working with, in which field you are, and then in which year. And then the program will, based on the design that you've chosen, create your randomization. The next step then would be putting these trial designs into a field, and we have a tool for that as well, which is this one. So on the left-hand side, you can see the outlines of your field. So you have to know how many plots you can actually fit into a field. And then on the right-hand side, you have all your trials, and then you can manually put your trials into the field in the way that you want to have them later on in the actual field. So another, just for you to see what the software looks like, here we have a little bit of, that's how you get your statistic analysis. And then we have several different ways to visualize your data, heat maps, box plots, and so on. And as I said before, just like the analysis and the randomization, also the visualizations are based on R. If you have something certain that you want to use, we're able to integrate this into the software. So to wrap it up for you, so EasyBreed supports the entire breeding process and has a molecular or modular structure. If users don't need all of the modules, they can just use certain ones and just also pay for certain ones. We can adapt the software to the individual needs of the users. For example, we are now working with a tree breeder in South Africa, and we adapt the program to his needs because tree breeding is, of course, a little bit different from cereal or other plant breeding. We integrate all the hardware that you have, not only from Wintersteiger, but also other companies. And as I said, with the example of tree breeding, we're actually suitable for all crops with this software. Thanks a lot, and if you have questions, feel free.
Is any question for? I had just one. What is the NIR system analysis? Plant composition or something like that?
You mean what the
NIR.
NIR, near infrared?
Yes, near infrared.
Yes.
So do you measure plant composition or?
Well, you have the band for, I have to be I would lie if I knew the exact wavelength that it measures in the near infrared spectrum, but you have certain wavelengths that you take up, and the NIR is mostly used to then afterwards calculate the vegetation indexes. I don't know if I got your question right, or it's okay.
I'm just confused. Thank you.
Okay.
So I think this section is over, and now. Oh, sorry, that's a question.
What are the advantages in terms of platform with regard other options like Mavic?
You mean the drone?
The drone and the embedded
Sensor. Well, the drone, as I said, is a fixed wing one, and there are copter drones in opposition to that. The fixed wing drone have the advantage that they are quite fast. So you can fly you can cover your trial pretty fast. Whereas with a copter drone, you need to invest quite a lot of time to fly over the trial. Then concerning the camera, well, I mean, it's a multispectral one. I don't think that right now in practical plant breeding we're at the point where we should already or can already use hyperspectral ones. So I think with this camera that's in this drone, you're at the moment very well equipped for everything that you want to do. One downside of this drone is in comparison with the copter drones, you have to fly at a certain level. And because the fixed wing drone has to fly at a certain speed, otherwise it will crash. So if you go lower and you're still flying at the same speed, your pictures will get blurry because you're too fast, you're moving too fast. And so with the fixed wing drone, you can't get such a high resolution as you can get with a copter drone. So that's the downside of a fixed wing drone. That probably with a copter drone, you will get higher resolution pictures. And I can also tell this, the resolution that we can achieve with the drone right now is perfectly suitable for all the traits to be measured that I've shown you. However, I will think in the future we will need higher resolution in the pictures for the algorithms that are coming, especially if you want to go into serial head counting. Okay, why did we choose that? Well, it's a little bit a tricky question. We actually also wanted to have a copter, and our customers should have the choice. But as I pointed out in the beginning, we're working with another company in a partnership, and they had the plan to construct a copter drone and never did it. And that's why we right now ended up with only the fixed wing drone, and we can't offer our customers the choice between a fixed wing and a copter drone.
Okay, thank you very much. We are closing this session, and thank you for your talk also.
Okay, so thank you very much for being here and your active participation. So now it's called in the program Eucarpia meeting, so don't worry, it won't be too long. It's a formal part of the meeting in which we will decide on the next steps, because it's very nice to be here all together, but we have to think in advance about the next steps. So the current situation is that, as you saw, Violetta here is the chair of the scientific committee and was in charge and did a great job to organize all this meeting. And so, yes, you deserve it. And my own role was very easy. It was to be I'm the chair of the section. So the tradition is that the chair of the section, after a meeting, gives, so how to say that? So let's say the proposition is that Violetta will become the next chair of the section, and myself, I will be a part of the scientific committee. It's the tradition in this section that the person who has been in charge of the organization of the meeting becomes the next chair of the section. And this has worked very well like this. I succeeded to Domagoy, who is in the audience. And there has been a single exception, is that after Munich, I continued because Christian preferred that we would do so. But we think it's good to come back to the initial practice, and we trust that Violetta will be an excellent leader for this community. So if everyone agrees, we will write down in the minutes that Violetta is now, since no, I will remain the chair until tomorrow noon. Okay.
Yes,
yes. But after tomorrow noon, Violetta will become the chair. So is it okay for everybody? Okay. So thank you very much, and thank you, Violetta.
Thank you, Alan, first for help that you gave me at the beginning in organizing this conference. Thank you, all of you, for your support and for coming here. And as I said a few days ago, I'm really happy that we managed after 2 years, more than 2 years, to have this meeting in person. And I do hope that you have a nice time and that you will enjoy it tonight. And thank you for your support. I will do my best to help at least in the same way like Alan did to next chair of Eucarpia and next organizer of Eucarpia meeting, which should be in 3 years. Okay. Alan, now it's your turn.
Okay, thank you very much. So the next thing, so thank you for this. That was the first part. The second part is about our next meeting. So the tradition also, and we think it's good, that this meeting would occur every 3 years. So it's now 3 years from Munich, and so it would be now 2025. And so we need to think about our next meeting. So of course the best is to discuss that a little bit in advance. So the proposition that we make in agreement with the people of the sites I will mention, I'm putting some, some tension in that. Tension. Well, I think it's no longer such a big secret. So the proposition is that the next meeting will be in Bologna. And thank you very much to Silvio and the colleagues for accepting to welcome us. So I don't know where Silvio is. I guess you will be, Silvio, our main contact for the... Silvio
but trust us, he
Silvio,
agreed.
Silvio is a great guy, and he will do a fantastic job. So next time, Bologna. And there is also a strong motivation to go to Pontevedra in the community in Galicia. And so the colleagues in Pontevedra, I'm thinking of Rosanna in particular, kindly agreed that they probably would welcome the meeting in 6 years from now. So is it okay to you to put this in these terms? Okay, so if no one is against that, we propose that we move forward with this agenda. Bologna is a very nice place. Pontevedra is a very nice place. The two places are very convenient also. So if okay to everybody, no opposition, please we proceed like this. If someone would be interested to organize the meeting in 9 years, so
please
you
let us know.
can think about that. There is no emergency. Okay, and maybe the final part is that if someone wants to say something about the main lines of the meeting, we can take a few minutes to do so. Maybe what I would just like to do is to recall that it's a plant section of Eucarpia. It's about maize and sorghum, and we are very happy to have both species represented. I think for the maize people, it's always very inspiring to hear about sorghum, and so and I hope it's also reciprocally true. And so there are all these species, and then you have sections, as was shown by the President Andreas Borner, on genetic resources, biometrics. So I want here to advertise the biometrics meeting that will be held in September in Paris, organized by Laurence Moreau and Tristan-Marie Huard in particular, and some young colleagues here are helping. So I think it's also very complementary because it's a good way to go deeper in breeding methodology, statistics across species, and there is a lot of information to be gained across species. Maybe the last things I would like to mention is that there is, as you see, the topic of Eucarpia was made very clear. It's about genetics related to breeding. So we find, and I think this meeting was really good from that standpoint, it's still very good, to find a balance between breeding, traits, and genetics, and so on. And for people who are more into, I would say, the molecular physiology, more genomes by themselves, there is a yearly meeting in Europe called the European Maize Genetics Conference, which gathers 50, 80, maybe people each year, maybe more, 100 sometimes, and which can be an opportunity for people to go deeper into the physiology, the genomics, and so on. That is here on one aspect of what we deal with. Okay, so my view is that the both meetings are complementary, and there is no reason to, I think, to absorb one or so the proposition is to continue like this. So if okay to everybody, the main lines of the next meeting should be in accordance with those from this meeting here. But this being said, if there are some burning questions or comments, maybe one thing is about the period. So it seems from the last meetings that this period of year is one of the best for the breeders. So, and also it's generally decently good weather. So,
yes.
and a nice time of the year. We are not too busy with our nurseries and so on. So that's a good time of the year. So we will continue. The format with this format of 3 days with a visit, I think it's appreciated in general by people. But if there is any question or comment on things that you think could be considered for the next meeting, please do so. We can take a few minutes. So if there is a comment, I'm looking. So if not, it's not at all a problem. We can. We have very nice perspectives for tonight. So we
Yes.
will meet at 9:30.
Not
We
9:30.
oh sorry
7:30.
7:30. Okay. Okay. So no question or comment. Okay. I don't want to frustrate anybody. So if so, I think we can consider that this section meeting was productive. So thank you very much for your attendance, and that we meet now at 7:30.
7:30, please. 7:30 in front of the hotel, and the buses will be the same like today. And the dinner for people who will not use the buses, please be at the place K. Yachting Club, as you can find on our website at 8 o'clock. And after that, the boat will start with 1 hour and a half. So on the River Sava and Danube. And I hope that you will enjoy this evening. Thank you very much for your support, for your attendance, and see you tonight.
Okay, thank you very much. Have a good evening.